All right, so moving on to the details of the central dogma, we're going to talk specifically and be able to describe the processes of transcription and translation today. So I'm going to have two separate videos on this worksheet, and you're going to do one on transcription, answer a couple questions on the Google form, and then you're going to do translation. So let's start out and just talk about what is the first process of the central dogma? What order do they happen in? What is the second process? Right. If you remember back to last class, the order that they happen in is transcription first. That's when you're turning DNA into mRNA, and then translation second when you're turning mRNA into your protein or your polypeptide. Okay. So the first step is transcription, like I just said. So when you go from DNA to messenger RNA, you're going to do the process of transcription. And that starts by unzipping DNA. So we know that DNA is two-stranded, right? So you have to separate those two strands with helicase. So helicase acts kind of like a zipper, and it unzips the two strands slowly so that you're actually going to be able to access the strand of DNA that you want to copy the mRNA off of, right? That instruction that you need to read, you have to open it so you can read it. So you're going to break the hydrogen bonds in between. And then one side of the DNA is going to be the template, and the other side is the complementary strand. And complementary literally just means matching. Okay, so the template strand, for example, if this is the template strand, it's going to be red, and the mRNA is going to match up with this strand. This strand on this side is the complementary one to the template one, so it matches it. So A belongs with a T, and then C belongs with a G, right? So they'll be matching together like that. That makes them complementary. Now, the template strand is going to be copied into an mRNA, which means that the mRNA is also complementary to this strand, which means that the mRNA is basically the same exact code as the complementary strand, except obviously U's instead of T's, right? So this picture kind of shows what it looks like when you have DNA and it's getting unzipped by helicase, and then one side is going to be copied from, and one side is going to be left alone, okay? Transcription begins at a place on the DNA called the promoter. So if you've ever been to the mall and you see people at kiosks in the center of the mall and they're handing out flyers and they're trying to get you to like talk to them about their product. They're like, "Hey, are you interested in, or would you like to try a sample of, or hey, we want to come in and look at the products that we have in the store." That's a promoter. It does the same job as a section of DNA where they're just trying to get you started and interested into something, right? It's just to catch your attention. So the promoter on the DNA catches the attention of the reader molecule, which is called RNA polymerase, and it's going to start reading after it accesses from the promoter point, right? It gets interested from the promoter, and then it reads from there on. An enzyme called RNA polymerase, like I just said, attaches nucleotide bases of mRNA to the template strand using the rules below. So the rules are as here. Like I said, if you have two strands of DNA that are separated, and this is the template strand, the mRNA is going to match the bases on the DNA using the mRNA version, right? In DNA, we have T, which is thymine, but in mRNA, we don't have T's. We only have U's for uracil, okay? So if your DNA has an A in it, the the RNA polymerase is going to build an mRNA U to match that one, right? It's going to complement it. So A is now paired with U in mRNA. T is paired with A. So if you see a T on the DNA, the RNA polymerase is going to go, "Oh, we got to put an A on there." If the RNA polymerase sees a C on the DNA, it's going to go, "Oh, we got to put a G on there." And if the RNA polymerase sees a G in the DNA, it's going to build a C onto that mRNA strand that is now growing because it's matching and being built based off that DNA template. Okay. Not all genes are expressed though. And I have to mention this because all of your cells have the exact same DNA inside. And the thing is, that's kind of weird to think about because it's not like your eyeball cell and your toenail cell look the exact same or even do anything close to the same function, right? But they all have the same DNA in them. So it's kind of weird to think about because why are they so different if they have the same DNA? And that's because not all genes are expressed. So eukaryotic genes contain regions that are not translated into protein sometimes. And these regions of DNA are called introns. And they get removed by a process called RNA splicing. And splicing just sounds like slicing because that's what it is. These things get sliced out and then they get removed and then they don't get turned into proteins. And that's normal. A lot of times in different parts of your body, certain genes don't need to be read because there's no function for them. So they'll get spliced out. And then the rest of the important parts that need to be read for that body part or for that body function for that cell, they're going to be translated into proteins And those sections that are actually expressed are called exons. Okay, think expressed and exons. So you cut away the introns by splicing and you express the exons into proteins. So why does transcription happen? Because we know DNA can't leave the nucleus. So something's got to leave the nucleus, and that's our messenger RNA. Where does transcription happen? Well, it's got to happen where the DNA is, so that's going to be in the nucleus. And that answers the next question of why it must happen there. How is mRNA made? It's made when DNA is split apart and then read, and an RNA polymerase will build the mRNA strand based on the DNA strand. So if you see an A on the DNA, you have to build a U on the mRNA. If you see a C, you have to build a G on the mRNA. If you see a G, you have to build a C. And if you see a T, you have to build an A on the mRNA. So they're matching complementary wise. They complement each other. Where does the mRNA go after it's made and why? It's going to go to the cytoplasm because that's where the ribosomes are, so you can build the proteins. And RNA splicing is when you cut out the introns and you only leave the exons to be expressed as a protein when the mRNA is going from the nucleus to the ribosomes. That way you're not making a bunch of extra protein sections that you don't actually need. And that way your cells can specialize in different functions because they have different proteins in them. So here is some practice now. So you're going to perform the function of, basically you're going to be doing transcription. And let's see if my text boxes will show up. Nope. Okay, so I'm going to insert some text boxes. And so we're basically going to go, hey, what is a letter on the DNA and what should we have on the mRNA instead? So if you see an A on the DNA, you should have a U on the mRNA to match. If you have a C, you should have a G. If you have a C, you should have a G in the mRNA. If you have an A, you should have a U. And the reason why we use U is because, again, there's no T, there's no thymine in mRNA. Okay, if you see a C, you should have a G. If you see a T, you should have an A should match with a U, and then a G should match with a C, and then a C should match with a G. So when you look at this overall, the DNA has been transcribed into mRNA because the A matches with a U, C matches with a G, G matches with a C, and then a T matches with an A. So this is our mRNA sequence based off of this DNA sequence. Okay, so I'm going to do one more here, just again to show. So if you start with a T in the DNA, you have to have an A that matches with that T. A G is going to match with a C. An A is going to match with a U. Remember uracil. A T is going to match with an A. An A is going to match with a U. Another A is going to be another U. A T is going to be an A. A G is going to be a C. A C is going to be a G. And an A is going to be one more U. Okay, so again, if you have DNA, this is the mRNA messenger that is going to be created based off of it, and then it's going to leave the nucleus and go to the ribosomes in the cytoplasm.